Good afternoon, everyone. And formally, welcome to 52nd Annual Meeting of American Pancreatic Association. As you know, as is our tradition, we had a pre-meeting this morning, which was very successful on pancreatitis. And now our formal meeting starts. And as is traditional, our first session always is Hirshberg session on pancreatic cancer. And of course, Aggie Hirshberg does not need any introduction. She has been on this podium many, many times. Everyone knows her. But still, I want to appreciate on behalf of our president, Dr. Hines, thank you for supporting this event. But more than that, thank you for doing what you do for pancreatic community every year, day in, day out. You have spent most of your life on this, so we really, really appreciate that. Let's do a big hand. Before we start, Aggie, maybe you want to come and just say a couple of words, and then we'll start the session. Please. You're good? I'm not the boss. I'm the servant for American Pancreas, but thank you. Maybe we'll have you tomorrow during dinner when we give Hirshberg Award, we will have you come on the stage. With that, I'd like to introduce again someone who doesn't need introduction, my co-moderator, co-chair, and our president, Dr. Joe Hine, who will introduce the first speaker.
Thank you, Dr. Saluja, and thank you, Aggie. Thank you so much. Our first speaker is Dr. Andrew Biancan, Regis Chair of Surgery at the University of Glasgow, a Cancer Research UK clinical scientist, a Wellcome Trust Senior Investigator, Fellow of the Royal Society of Edinburgh and Academy of Medical Sciences, and the Director of the Wolfson Wool Cancer Research Center. Dr. Biancan will be speaking to us on precision oncology for pancreatic cancer.
Good afternoon, everybody. Thank you for the honor of speaking at this year's Hirshberg Symposium for the American Pancreatic Association meeting. Apologies, I can't be there in person. I'm disappointed not to be there to meet again with friends that I've known for a very long time, colleagues, others, after such a period of not being able to see each other. After my recent bout of COVID, I was told that I probably wouldn't get in based on a PCR test. So hence, here I am at home. So today I'm going to talk about what we've been doing in our effort to strive forward with pancreatic cancer. I'll just share my screen, if I may. There we go. So hopefully you're getting an image of our university there in the bright sunshine. Though I'd like to say that happens quite often. Doesn't, but when it does, it's glorious. So here are my disclosures. So this is a slide that's been around for a while. Margaret Tempera kindly passed it on to me a few years back, originally made by Philip. And I think back then it was back in 2014, 2015. Not a lot has changed. Well, what's particularly dramatic is that we now have more and more drugs to choose from. Most of them are either incrementally effective and are often not used, like erlotinib, or they target very small subgroups of patients. We don't know ahead of time. But apart from perhaps pembrolizumab and olaparib, where we can look at germline mutations or tumor mutation burden or mutations in BRCA1 and BRCA2, however, that's only germline. We haven't even looked beyond that into the somatic aspect of things. And we start to get a bit stuck. We have all these drugs to potentially choose from. We choose on the ones that's most likely to work, but is it the right drug for that individual patient? If we look at other examples of cancers where the approach of matching the right drug with the right patient will work, think of breast cancer with tamoxifen and its derivatives, targeting estrogen receptor positive breast cancer. Not only is that the case, but also we know so much more about the molecular pathology of breast cancer after treatment. We know that We know what the patients look like molecularly when they come into the study. We know the mechanisms of resistance afterwards. We know when to position new therapeutics. For example, we're seeing in new developments that post-PI3 kinase alpha therapy and post-palbociclib therapy, there's a coaptation or focusing of mTOR-mediated resistance, which gives you the opportunity to position a therapeutic. Pancreatic cancer, as with many cancers, I suppose, is very far behind that. And that's where we want to be. What do we know? This is just our data parcel, but many others have presented and demonstrated similar things as well with regard to the molecular pathology of pancreatic cancer. If you look genomically, we still have those 4 mountains, KRAS, P53, P16, and STAT4. We have a few hills, around 10 or 15% prevalence, and then we have an ocean of infrequently mutated genes. It makes it difficult to target. Something about that combination of those 3 or 4 major drivers makes pancreatic cancer very difficult to drug. We can also start grouping things in different ways. There's structural character taxonomies of the genome. We can see an example there, some unstable genomes where there's some DNA damage response deficiency, and that gives us some insight, but not a lot of clinical applicability apart from that unstable genome example. We can see mutational signatures where we can understand potential causes of a cancer, and some, again, like the big signature 3 or the BRCA signature can be potentially targetable, although we can't detect that in the clinic yet. It takes a whole genome to detect that, which we can't do. Or we can start grouping them into functional elements, into mechanisms, etc. Just give them, it's a chart there. And many of these things are published and people can read them. But the genome gives us some avenue in. Now, when we get richer data is in the transcriptome. And in the transcriptome, because of the density of data parcels, you can start to split things up more into subgroups. It's not just a discrete, sparse data set of mutated or not mutated. You have various degrees of expression. And so this is our contribution. There are many others, slightly different characteristics, slightly different nomenclature. In these early days, we still need more data. But what is clear is that there are 2 lineages that emerge, one that looks like the pancreas and one that doesn't even look like endoderm. And that one that doesn't look like endoderm is associated with a poor prognosis. So if you summarize this and bring together some of the data parcels that are out there, we see these 2 clear lineages. And that's not surprising, because what we see in these 2 lineages, the biggest difference is epigenetic regulators, mutations in epigenetic regulatory genes, which shut down a lot of transcripts. So there's little wonder that the transcriptome gives you such an enormous separation when you're clustering. But as we start to delve deeper, and we see some nice work coming out of Canada with Faisal Notta's group, we're really delving into discovery of in-depth analyses of different potential subgroups. But we see within these subgroups, we can see some opportunities where we see immune clustering, differences in immune subtypes between the 2 in the microenvironment. And one study we looked at was we generated a number of cell lines in collaboration with back then the Diane Siemian, others at the Mayo, really putting together about 40-odd cell lines. And when we looked at these cell lines, even though we accepted that this does include the microenvironment, we get some information, but we have to interpret it carefully. What it did tell us is, because we didn't have any normal tissue there, dramatic differences between the epithelial components in specific pathways. These are just morphogens, but we see big differences in signaling pathways. So it's little wonder, if you're doing a clinical trial, you're not separating squamous from classical, or basal-like from classical pancreatic. They could be doing completely different things. They could be, even promoting some aspects of one subtype, whether it's inhibiting some of those. And we published some of those examples, and then opportunities of how you can use these subtypes that may cluster together with things like replication stress and other aspects that may enrich for potential responders in clinical trials. Of course, if you map out this as an actionable genome, and there's some great work from Know Your Tumor, there are opportunities there. Have we tested them appropriately? Probably not. But I think precision oncology for many cancers needs to go beyond just let's find a target and give them a drug and see if that works. We need to think about the breast cancer example that I've mentioned. We need to understand what is the molecular pathology of our patients and their cancers when they come in through the door. How are they different from being early resectable or from metastatic? Which are the ones that actually get onto trial? And then as we build on that, what happens to the tumor as it evolves after its first line of therapy, its second line of therapy? What are the mechanisms that are at play? Otherwise, we're still very much in the dark. So in an attempt to do this, we built a platform called Precision Panc. And moving from a single trial to trial platforms where we can start to build these bits of information of molecular pathology and also offer patients clinical trials. And then someday get into the realm of real world data where we learn from every patient rather than 1 or 2 or 3% of them when we run clinical trials. The goals or questions Precision Panc aims to address is that how can we use current treatments better? It's clear that we can better select patients for these treatments. As I mentioned, how does pancreatic cancer evolve with time and with treatment? And how can we accelerate therapeutic development through a platform and then potentially utilizing real-world data? We built 3 pillars of Precision Panc. One was the discovery pillar, bringing together data sets that can be mined to prioritize strategies, which then were tested in the preclinical world, preclinical models, etc., by Alan Sampson at the Beatson and others around the UK and collaborators around the world to build a preclinical platform of evidence that would give us sufficient confidence to take something to an early clinical study based on a biomarker hypothesis. And of course, during these studies, we'll accumulate more information to feed the discovery pillar. We had to build a sequencing laboratory back in the day. Now this laboratory has become almost purely clinical. It's moved to the hospital system. It is now finalizing MHRA, your equivalent clear regulatory approvals, and we'll be delivering molecular tests in the clinical world starting probably January 2022 for not just pancreatic cancer, for all different cancer types. So in order for us to do this, we had to build an affordable and fit-for-purpose genomic test that really integrated seamlessly with the healthcare system. So you didn't have to take more or additional biopsies. Of course, in pancreas, we never really took biopsies apart from brushings in the past, but now we can do this on a 21 gauge. It's sort of halfway between a fine needle and a core, either percutaneous or through endoscopic ultrasound. And as I'll show you, the vast majority, the success rate is over 80% from intention to biopsy and get it to a result itself. We built the next evolution of tests built on beyond the exome itself, using ICGC, pan-cancer analysis, whole genome data to look at beyond small variants, looking at copy number, structural variants, signatures, fusions, immunological resistance mechanisms, regulatory regions, and also some sample integrity components. And we've called it the Glasgow Cancer Test. And it's available globally through our distributors, Agilent, and it's extremely cost-effective when delivered on its own. Every patient in Precision Panc gets a report characterizing their particular individual mutations, and as they enter Precision Panc, this is the master protocol, and probably the most significant innovation was to intervene with patients early. Don't ask them about a clinical trial when you're about to make a treatment decision and they're going to need additional biopsies. We interact with our patients very early, just with a provisional diagnosis on a clinical grounds. And so when they go for their diagnostic journey to get their diagnosis, we parallel the research journey, the clinical trial journey. So when it's time to make a treatment decision, the molecular profile is already done and the clinical trial options are on the table. There's no further need for any biopsies and other things. We try and have every patient have an opportunity on Precision Panc. So it's the first thing a clinician thinks of, to cover the various indications and stages of treatment, neoadjuvant, locally advanced, first-line metastatic, second-line metastatic, and we're building out this portfolio so every patient has an opportunity for a clinical trial. So if you looked at this schematically, so the master protocol sort of intervenes between different lines of therapy, and not only can you enter Precision Panc through the original master protocol, through Primer 001 and others, you don't have to participate in those studies. You can elect to have standard of care, but you still get a biopsy, you still get a report and a result, and you're still potentially eligible for second-line studies or off-label therapies if that's an option. And you can even come in externally after first-line standard of care treatment, come in and for second-line studies. You can see the portfolio we're building out there. The second-line umbrella is where the more targeted studies occur. The first line is more biomarker discovery, biomarker validation. And so, for example, in Primer 001, there's interim analyses built in to test biomarker-driven, biomarker-associated responsiveness of platinum therapy when you're comparing FOLFOX-A versus Abraxane. Gemcitabine is the hypothesized biomarker. It's a structural variant signature associated with response to a platinum therapy in pancreatic cancer. And that allows all patients to have an opportunity to enter Precision Panc. Where are we up to now? We're starting to nudge 500 patients entering the study. We get some attrition early on because when we discuss this with patients earlier, the diagnosis isn't finalized. It may well be ampullary or some other cancer, so it doesn't ease in pancreatic, so some patients are lost there. 369 get registered, they've had their biopsies and they get molecularly profiled, but we can see the challenge of pancreatic cancer with significant attrition. And we study this carefully to understand where we can do things to change the degree of attrition. See, from 369 patients, 208 go into study. So that's like a 30–40% attrition rate. And some we look at avoidable and unavoidable. So while some decline the trial for various reasons, if they've still had a biopsy, they've got a result, they can still enter in the second line. And that's the signal we're sending to our clinicians, is that even if you don't like the trial, you're part of the platform, there are trials in second line for you as well. What about the molecular pathology of pancreatic cancer? In the metastatic advanced setting. So it is very similar to the early disease, particularly with the four mountains, KRAS, p53, p16, SMAD4. But there are also some key differences. If you compare this to whole genome sequencing data and historic data in early disease, you can see the darker bars, the bluey black bars are those that we see in metastatic disease, and the ones on the right are the ones we see in early disease. And you can see that there are some differences in some of the mutation prevalence. And this is particularly interesting because these mutations are associated with the better prognostic classical pancreatic subtype versus the poorer prognostic. And so you are starting to enrich for these in the later disease setting because your poorer prognostic cancer patients are dying off. So whilst early on you would imagine there would be a greater preponderance of squamous disease in early advanced disease, then because of their rapid attrition, the proportions you would imagine would change. We asked the question, what about actionability for the trials we currently have ongoing or planned for the platform? And it's about 30% of patients or more would be eligible for a targeted study based on the targeted studies that we have opening currently. And this is based on the first 150, 160 patients. And the denser data set was more like 260, 280 patients. And we see, what about KRAS? We don't see G12C, but we see mostly G12D and V, and you can see those there. But what's really interesting is in the KRAS wild type subgroups, we do see alternative drivers. In fact, we probably won't call something wild type because of the challenges in lower cellularity cases, in that unless we see an alternative driver. What about the transcriptome? So these are data from a technology called TempoSeq from BioClavis, works on a section of formalin fixed paraffin embedded material, just one section, and you get these great data parcels. We weren't sure at first, so we kicked its tires and tested, and this is the data parcels that you get. So it's not quite as good as RNA-seq, but RNA-seq on formalin fixed paraffin embedded little core biopsies, it's pretty tricky. So if you can do it at all. So we see the same clusters, the lineages and the clusters. What's interesting, just as I alluded to earlier, we can see that those patients that aren't fit or there's attrition, there's a higher representation of squamous patients in that group, representing the poorer prognostic prognosis of that particular group itself. So I'm just going to finish with what do we do with these data? I mean, it's great that we publish these data and then we put these data out in the public domain, maybe in an Excel spreadsheet. People may or may not see it. So ICGC and TCGA have done a great job. There are more data parcels growing. Look at AACR Genie. But how do we share these data in a meaningful way? So this is across all cancers, and it's the next iteration of the International Cancer Genome Consortium called ARGO, Accelerating Research in Genomic Oncology. And our goal is to deliver 1 million patient years of precision oncology knowledge to the world. And that exemplifies the importance of longitudinal data. 200,000 patients with 5 years follow-up, 300,000 patients with 3 and a bit years follow-up. That's the principle of understanding things longitudinally in patients as they undergo treatment. And to date, we have, this is a little bit older slide, we have 15 or 16 countries now. We're about halfway there with promised participants. The portal is open. We have about 5,000 whole genome sequences going up that are up there on the portal. The clinical data isn't as well as we'd like, but 1,000 of those have really nice tight clinical data. For a project that's aimed to finish in 2030, we're well underway to getting the number of participants we need. And to address these sorts of questions that we want to know as clinicians and our patients want to know for an individual, not on average. So there's a lot of people that contribute to this. It's great to be able to get up and speak about it, but these are the main people of Precision Panc and our GPOL activities. There are hundreds of people around the UK that contribute, and these are our funding bodies. And thank you very much. And although I had to record this session, I will be in person at the Q&A session later tonight, about 9 or 10 o'clock or 11 o'clock my time. So I look forward to it. And thank you very much for your attention, and look forward to speaking soon. Bye.
We will have Q&A at the end of all the 4 talks in this session. It's a pleasure to introduce our next speaker, Dr. Laura Wood. Laura is an associate professor and director of GI and liver pathology at Johns Hopkins. She got her MD and PhD at Hopkins, and for her research, she worked with famous Dr. Bert Vogelstein, and she led the first, during her PhD, she led the first whole exome sequencing study in human cancers. And after completing her residency, she did a fellowship in GI and liver pathology there, and now she runs a very active translational research lab and have all kind of 3-dimensional and 2-dimensional models to find some new cures, I hope, for pancreatic cancer, something we very badly need. Welcome, Laura.
Thank you so much. Thank you so much to the organizers for inviting me, and then to all of you for showing up. This is my first in-person meeting since February 2020, and it's really great to be able to share it with you all. So we're going to shift gears a bit. I think Andrew's talk provides an amazing framing for the topic of pancreatic cancer genomics. And actually my slide first slide introduces some of the similar concepts. So now it's been more than a decade since we got the first look at the genomic landscape of pancreatic cancer. And Andrew mentioned the 4 mountains. These are the most frequently mutated genes, mutated in the majority of pancreatic cancers. This is KRAS, P16, P53, and SMAD4. But then the landscape is really dominated by these infrequently mutated hills. And this was 13 years ago. But now, as Andrew mentioned, there have been hundreds and hundreds of exomes and genomes analyzed. The hills are still the hills, or sorry, the mountains are still the mountains, but the hills have really highlighted different groups that are amenable to specific targeted therapies. And again, that was the subject of the previous talk, but what I'm gonna do is focus more on how do we understand initiation and progression in pancreatic cancer? Those pancreatic cancer studies are at the advanced stage. So how do we understand what happens in the precancerous setting? So we can infer this from sequencing data from advanced cancers, or we can directly analyze the precancerous lesions. And from the title of my talk, I'm sure you've guessed that I'm going to talk about directly analyzing these precancerous lesions to understand what happens before pancreatic cancer becomes malignant. So these precancerous lesions come in 2 flavors. The majority of pancreatic cancers arise from microscopic precancers. These are called pancreatic intraepithelial neoplasia, or PANN. These are microscopic, so less than 5 millimeters, which means that they're not visible in imaging studies, and they're not visible in a gross pancreatic cancer specimen at surgery, like in the pathology suite if we're looking at it. These are graded based on the morphologic atypia of their lining epithelium. So low-grade PANNs are very, very, very common. They have minimal atypia. Then high-grade PANNs are much less common. They have severe atypia, and they're the step kind of right before invasive cancer. An important minority of pancreatic cancers, though, arise from these larger cystic precancerous lesions, the most common of which is intraductal papillary mucinous neoplasm, or IPMN. These are by definition bigger than a centimeter. They are often much larger than that, which means that they're visible in imaging studies. They're also visible in a pancreatic resection specimen, which means we can harvest tissue from them. And because they're diagnosed as larger cysts, they often get resected before they've progressed to invasive cancer. So they provide a really nice opportunity to study precancerous pancreatic neoplasia in human samples. Like PANNs, IPMNs are graded based on the morphologic atypia of their lining epithelium. Low grade is super common and low risk for progression. High grade is much less common and much higher risk for progression. So now I want to circle back to the serpent's egg analogy. So this came from my friend who's a pancreas surgeon in Lisbon. His name's Jorge Paulino. And he suggested that this would be a really great way to frame a talk. And I was embarrassed. I had to email him back and be like, I have no idea what this refers to. And he said, oh, it's this amazing movie by the Scandinavian director Ingmar Bergman. It's one of my favorite films. Anybody who knows me knows I have small kids. So this is the closest to Scandinavian film that I get in my house. So I haven't seen that movie, but when Jorge suggested it, I went and looked at the literature. Not the scientific literature, but like literature. And it turns out this analogy kind of permeates through a lot of plays and books. I put a quote here from Shakespeare where it's mentioned in Julius Caesar, and therefore think him as a serpent's egg, which hatched would as his kind grow mischievous and kill him in the shell. Meaning once pancreatic cancer hatches, it's a snake, it's gonna eat, it's gonna cause damage, but the precancerous lesions are the eggs, and can we kill them in the shell? So that's our question here. So the precancerous lesions, again, these are the eggs. The early invasive cancer is the snake just hatching. And then once the cancer grows large, the snake does a lot of damage. So can we kill these precancerous lesions in the shell? I would argue that we can, but there's 2 things we need to do. First is we need to find the eggs. And the second, which is much harder, is to predict which ones are actually going to hatch. Because the vast majority of precancerous lesions are never going to progress to invasive carcinoma. And so there's a huge risk of overtreatment. So with this, I'm going to talk through some of the studies that we've done. I'll focus mostly on IPMN because, as I mentioned, they're larger. They get resected before they've had an associated cancer. And so they're a great opportunity to study premalignant tumorigenesis. But I will circle back to PANNs at the end. So I'm going to tell 3 short stories about IPMNs and their genomes. I'll focus first on the polyclonal origin of IPMNs, and I'll talk some about later tumor genesis and malignant progression. And then finally, I'll circle back to a new driver gene. So the work on polyclonal origin was the work of a now graduated PhD student in my lab, Kathy Fisher. And what Kathy's goal was initially to assess genetic heterogeneity in IPMNs. So she took 30 surgically resected IPMNs, 20 that were low grade and 20 that were high grade. And because IPMNs, when they're resected, they get entirely submitted for histologic examination and pathology to exclude the presence of an invasive cancer, which means that for these resected lesions, the whole IPMN is sitting in paraffin blocks in the pathology archives. And so what we did is we took all of the blocks from these 28 surgically resected IPMNs, we separately microdissected them. This gave us 233 total DNA samples. And then we did targeted sequencing of 15 genes known to be important in pancreatic tumorigenesis. And this is, kind of a very broad overview of Kathy's data. And in this, each group of bars in the columns represents one IPMN, with the low grade on the left and the high grade on the right. Each tiny little bar in there is a region of that IPMN, and then each row is either a mutated gene or a mutated position. So in this graph, KRAS and GNAS, these key oncogenes, each mutation is split out into separate rows. And what I want you all to take from this very broad picture is that the low grade is different from the high grade. So this side on the left, when you look at KRAS, you often have multiple different KRAS mutations in the same IPMN and even in the same region. And there's often not a single KRAS mutation that's shared among all of the regions in an IPMN. So when you look at the red up at the top, you see lots of little bars kind of scattered all over the place. In the high grade, what you see is there's a single KRAS mutation that's shared among all regions in the high grade IPMN. So there's much less heterogeneity in the high grade regions. We quantified this and statistically compared them with Jaccard similarity coefficients and showed that with respect to these initiating oncogenes, the low grade IPMNs are more heterogeneous than the high grade. What we were really interested in now that we had this observation was, what does this mean for the origin of IPMN, that these low grade IPMNs have such heterogeneous KRAS mutations? So our first way to look at this was to do computational reconstruction of the evolutionary history. We worked with Rachel Karchin, who's at Hopkins. She has an algorithm called Schism that we use to reconstruct the evolution of IPMNs from the sequencing data. And what we found was that when there were multiple KRAS mutations in one IPMN, they didn't share a common ancestor other than the germline. So this suggests that these really represent independent clones in that IPMN. But the challenge with this data is that it's targeted sequencing. And so we couldn't exclude that there was some other mutation somewhere else in the exome that they shared that we just didn't sequence in this set. So the way that we addressed this is we did whole exome sequencing on 2 regions with discordant KRAS mutations. So these are 2 regions of the same IPMN that had different KRAS mutations. And when we did this, they never shared any other mutations either. So again, this really highlights that when you have these 2 separate KRAS mutations, they represent independent clones. And we in a separate project did single cell DNA sequencing from IPMNs, and in a couple IPMNs we did find multiple KRAS mutations in this project. And always when this happened, they were always in separate cells. So this again goes with the theme that these different KRAS mutations really mark independent clones. So from this clonal evolution study, we showed that there's heterogeneity with respect to the early driver gene mutations, and it's more prevalent in low-grade IPMNs. These mutations occur in different cells, and from some in situ hybridization data that I haven't had time to show, they are spatially separate. This suggests a polyclonal origin for a subset of IPMNs. But what can sequencing tell us about this transition to invasive carcinoma? So we'll talk a little bit about genomic analysis of malignant progression. This is the work of Michael Noe, who is a pathologist and did his PhD in the lab. So Michael's goal was to compare the somatic mutations in the non-invasive IPMN and adjacent invasive carcinoma. So what Michael did is he separately dissected, either macro or micro dissected, the IPMN and then the cancer that was immediately adjacent to it in the same tissue block. And then he did whole exome sequencing of these separate but adjacent areas of IPMN and cancer. And then in 7 of these patients, he pulled all of the blocks and did targeted sequencing of all of the blocks to really reconstruct the evolution. One of the findings of this study is that as kind of has been suspected clinically for many years, IPMNs really represent the evolutionary precursors to invasive cancer. In all of the cases where we had many samples, we could reconstruct the evolution and show where the cancer arose from the IPMN. When we look at the mutated genes, the top three, KRAS, P53, and P16, these are the three of the mountains. They were mostly shared between the invasive cancer and the adjacent IPMN. But SMAD4 was different. It was the only gene that had multiple mutations that were limited to the invasive cancer component. And there's an example here on the left with the IPMN in blue and the invasive carcinoma in red. And they share a KRAS mutation, they share a P16 mutation, they share a P53 mutation. But this SMAD4 mutation, which is nonsense, is only in the invasive cancer and is not in the immediately adjacent IPMN. And we saw this pattern in 4 IPMN cancer sample pairs, which suggests that in at least some IPMNs, the SMAD4 mutation is the thing that drives it to invasive carcinoma. But just as striking, many IPMNs actually lacked a driver gene that was only in the cancer, suggesting that there's something other than somatic mutations that can mediate this transition in some patients. So the next most commonly mutated gene was RNF43. This is a gene that was first discovered in IPMNs, and it's very frequently mutated in IPMNs, far less frequently in invasive cancers that arise in the absence of IPMNs. But it had the opposite mutation pattern. Whereas SMAD4 was enhanced or frequently mutated only in the invasive cancer, RNF43 was often mutated only in the IPMN and absent in the invasive cancer. So it arose in the IPMN, but the clone that eventually invaded lacked the RNF43 mutation. And it had this really striking pattern that no other gene had, where there were multiple mutually exclusive RNF43 mutations in the IPMN and each only in a small region. They were all inactivating, frame shifts and nonsense mutations, and each only in one small region. We also saw this again in our single cell sequencing data. This was single cell DNA sequencing of a separate cohort of IPMNs. And when we found RNF43 mutations, there were often 3, 4, 5 of them in an IPMN, and each limited to its own small subclone. So these RNF43 mutations, they occur in subclones that don't give rise to invasive cancer. And there's convergent evolution where you have multiple mutations with distinct subclones, multiple mutations in the same gene in distinct subclones. And this suggests something really unique about the evolutionary forces that are acting on RNF43, and we don't really know the mechanism for this. So from these studies of malignant progression, we've shown that IPMNs are precursors to invasive pancreatic cancer. These SMAD4 mutations may drive malignant progression, and the RNF43 mutations are heterogeneous and often limited to subclones that don't invade. So the final part of IPMNs I want to discuss with you is this new oncogene that we've discovered in IPMNs, KLF4. This is work by 2 pathologists in the lab who are postdocs, Kohei Fujikura and Waki Posada. So their goal was to analyze the transition from low-grade dysplasia to high-grade dysplasia in IPMNs. And to do this, they did multi-region whole exome sequencing of low-grade and high-grade matched samples from IPMNs. So they took 17 resected IPMNs. They're both pathologists, so we looked at all the slides. We specifically picked cases where there was sizable areas of low-grade dysplasia and high-grade dysplasia in the same IPMN. We then did laser capture microdissection, separately isolating the different grades of dysplasia, and then did whole exome sequencing. And so it was between 2 and 6 regions per IPMN, 76 total neoplastic regions, all were sequenced whole exome sequencing. So when we look at the mutated genes, the top 3 in the list are ones we've heard before, KRAS, GNAS, and RNF43, but KLF4 had not been previously associated, these somatic mutations in pancreatic tumorigenesis. And they had a really striking pattern, which is that they were enriched in the areas of low-grade dysplasia and often not present in the matched high-grade samples. So these KLF4 mutations, in addition to that interesting pattern, they occurred at one of 2 hotspots. So two codons in the gene, codons 409 and 411. When we went back and looked at all the other IPMN sequencing data, we did find a couple cases that had these KLF4 mutations. They tended to be buried in the supplementary materials. And I think these weren't discovered as frequent because most IPMN sequencing studies have focused on high-grade dysplasia. These are very rare in PDAC, and they occur at critical residues in the DNA binding domain. Interestingly, they've been reported in a subtype of meningioma. the same codon 409 mutations, suggesting again they're functional, but we don't know at all what they're doing in pancreatic tumorigenesis. We did confirm that these KLF4 mutations can be detected in cyst fluid. Again, that they're more prevalent in areas of low-grade dysplasia, but they still occur at decent prevalence in high-grade dysplasia as well, suggesting they're not going to be a standalone biomarker for low-grade IPMN. So these are common, they're limited to low-grade regions, and the presence of KLF4 could suggest that the egg is unlikely to hatch, though again, it's not kind of strongly enough correlated to be a standalone biomarker. So taking all this together, I think we need to revise how we think about the evolution of a pancreatic cancer. So it's not a simple multi-step tumorigenesis model. It starts with polyclonal origin, then we get these KLF4 mutations that are in low-grade subclones only. We have convergent evolution with respect to other mutations. And then finally, we get invasion or transformation to invasive cancer, which can be driven by SMAD4 loss, but is often actually not driven by a somatic mutation. So how can these findings improve the care of IPMN patients? I think we need to use them to improve preoperative prediction using cyst fluid genetics. We do have one new genetic biomarker, KLF4, but as I said, that alone is not gonna be a single biomarker because it does happen, albeit at lower frequency in high-grade IPMNs. So we could use combinations of genetic markers to determine cyst risk, also incorporating the number of mutations in a given gene or the variant allele frequency rather than just a binary mutant versus wild type designation. But any of these are going to require testing and validation in large cohorts of cyst fluid samples before we can implement them in clinical practice. So I promised to circle back to PANNs. And so the big question now is, the heterogeneity in IPMNs is really interesting, but do these same concepts apply to PANNs? So PANNs are microscopic, which means they're often discovered incidentally in grossly normal pancreas, and they often extend along the duct system such that it's really hard to understand how different parts of a PANN relate to each other. And so to really study these rigorously, we needed a 3D reconstruction approach so that we could tell within a tissue block what PANNs were connected and what PANNs weren't. And so to do this, we partnered with some folks in the School of Engineering at Hopkins, Denis Wertz and Pei-Shin Wu, and their graduate student Ashley Keeman, and we developed this approach called Coda. And what Coda is it's a deep learning algorithm that can automatically annotate and 3D reconstruct human pancreatic tissue and then quantify the different tissue types at the single cell level. And this is showing a normal block of pancreatic normal block of pancreatic tissue. The duct system is in blue, and then it's bringing in all the different tissue types in different colors. We did the islets and the blood vessels, and now we go down to single cell resolution. And what Coda allows us to do is to take a tissue block and then annotate all of the PANN within that tissue block and understand how they're all connected. And so we've done this. Now I think there's 15 different chunks of pancreas on here, each from a different patient. The dark blue is the normal duct, and each other color in each of these images is a separate, anatomically separate PANN. And when we saw these, we were really shocked as to how many PANNs there were in these relatively small, when you think of the proportion of the pancreas. These are, 1 centimeter blocks of tissue, and they would have, 10, 20, 30 PANNs in them. So the burden of PANN and the multifocality of PANN, I think, is much more than we previously anticipated. And now what we're working on doing is figuring out how to layer in genomic analysis onto these 3D models. So we can serially section the block of pancreatic tissue, and then we can annotate the PANN, do the 3D reconstruction, and then separately isolate the DNA from all these PANNs and actually sequence them and map those genetic alterations back onto the 3D models. And that's what we're working on now to really see how these IPMN concepts apply to the PANNs. So with that, I'll just wrap up to say that I think we can kill the serpent in the shell, but the key is predicting which eggs are likely to hatch to avoid overtreatment. And I would argue that studying the egg itself, studying the precancerous lesions is really critical to developing these strategies so that these molecular tuning strategies can play a role in risk stratification in patients with pancreatic cysts and pancreatic cancer precursors. I tried to highlight the folks from my lab who did the work as I went through the talk, but our lab is super collaborative, so I'd like to put up this slide just showing all the folks who have contributed. And then just to highlight our funding sources, and then I'm happy to take questions. I think that will be in the panel discussion. So thank you.
Thank you, Dr. Wood. That was a wonderful talk. Our next speaker is Dr. Diane Siemian. Dr. Siemian is a surgeon and a scientist. She is a professor of surgery and pathology at NYU Langone Medical Center. She's also associate director of the Translational Oncology Program at the Perlmutter Cancer Center and director of the Pancreatic Cancer Center there at NYU. She's past president of the Society of University Surgeons and of this organization, the American Pancreatic Association. She's a member of the Institute of Medicine of the National Academy of Sciences and serves on the Scientific Advisory Board for the NCI Pancreatic Cancer Task Force. Dr. Siemian.
Thanks, Joe. It's really wonderful to be here. I must say, just before I start my talk, I want to just again pay a personal tribute to Aggie for all the wonderful things she's done for our field. And then also just want to comment on the value of this organization has been to my career. I think I came to my first APA meeting in 1992. I was brought here by Craig Logston and John Williams, and both retired or almost retired. I think John is floating around here somewhere, but they really opened my eyes to the pancreatic community through this organization, and it has really been critical to the growth of my career. So I just want to make that point before I get started here. So I'm going to follow Laura's theme and talk a little bit about early detection. As a clinician and surgeon that's taking care of a lot of patients with pancreatic cancer, obviously I have the opportunity, as many of you do who are clinicians, to take care of patients in which we can make advances and improve survival by resecting tumors. And this graph on the left is probably familiar to many of you. It shows SEER data. The raised curve there on the left is resected patients with stage 1 disease. And of course, lower stages have worse survival. But even with stage 1 disease, that includes cancers that are as large as 2 centimeters in size. And you can see even with that, in patients that you can have negative margins, you're still looking at about a 40% survival rate. Just want to highlight some nice work that has been done by our Japanese colleagues, where they had the opportunity through screening programs to identify subsets of patients that had pancreatic cancers less than 1 centimeter. And with that data, the 5-year survival rate can approach 75%. And so what I'm going to do is make a case that as a field, it would be important for us to really shift in a major way our thinking about the emphasis we put on really going after early detection. And I'm going to highlight the potential role of genomics in getting us there. So just to put it simply, there's, the older that we get, the less we know. And there's still 3 basic questions we don't know. Who's at risk for pancreatic cancer? Every time I'm in clinic and I see a new patient with pancreatic cancer, about half of those patients, I cannot tell you why they got pancreatic cancer. I can't pinpoint a risk factor that they have. They might not have a family history. They're not smokers. They're not obese. That still is a conundrum. For patients that we know are at elevated risk, we still don't have strong data to tell us what that risk is. And then lastly, even for folks we know are at elevated risk, we don't have good paradigms to do much about it. We're still really struggling to figure out how to best either detect cancer early or optimally prevent this disease. And I would say of all the things we do, and of course, we also do a lot of work in therapeutics, but I think if we really can push early detection, that's the most powerful tool we can have to change survival. Now here I show things we know, and we know that there are germline mutations in these set of genes that put people at increased risk for pancreatic cancer, all of different relative fold. And in total, I think we know about 15 or so genes that are associated with increased pancreatic cancer risks. Many of them associated with other cancer risks, and they're listed here. It's also important to realize, and this has really just happened in the last couple of years, that we have an increasing appreciation of the fact that patients who have pancreatic cancer have germline mutations where you wouldn't have thought they had them. And I show data from 3 groups. The first is ours, and this was actually a project we started back at Michigan before I moved to NYU, where we got Myriad to pay for 500 consecutive germline tests and found that irrespective of family history, that I should have brought my glasses up here, almost 9% of patients had a germline mutation. And you can see we also have very nice work by our colleagues at Dana-Farber, UPMC, and other colleagues around the world that saw similar kind of numbers. And importantly, 50% of these patients, if you just took a family history, you would have never guessed they carried a susceptibility gene. So I think this data began to make the case that perhaps we should offer germline testing to pancreatic cancer patients. Now, while you have this data and actually making it happen, it takes a little bit of work. And we actually reached out to ASCO to assemble a committee to try to establish guidelines that all patients with pancreatic cancer should get testing. And it was an interesting process because as a committee, we put that forth, but then when the manuscript was reviewed, the reviewers came back and said, well, we actually think this is a good idea, but we don't really support it because it's too hard to do. We don't have systems in place to do this. To which several said, well, just because it's hard doesn't mean we shouldn't do it. And so you can see the title of this paper says provisional clinical opinion. And I couldn't get them to say guidelines, but after we got this paper out, I turned to my good friend, Margaret Tempero, and I said, Margaret, take the baton and have the NCCN, evaluate this. And you can see that shortly thereafter, the NCCN came out with formal guidelines that all patients with pancreatic cancer should get genetic testing. Now, amazingly, even though, and also to make the point, and you'll hear more about it, Andrew made the case, and Talia Galan, one of my colleagues, will talk about the fact that knowing this information can actually be helpful for the treatment of pancreatic cancer patients, especially if we find, for example, they have BRCA mutations, increasingly other mutations like ATM mutations. And then importantly, that helps you find people in that family that should also be screened. Also, just want to highlight facts of new information. Just so you know, there have been recent broadening of our testing criteria for unaffected relatives. Shown on the left is the fact if you have a single first-degree relative with pancreatic cancer, now the NCCN guidelines recommend testing. And then actually the genetic counselors at our place just point out the fact out on the right that for patients with breast cancer that have any close relative with pancreas cancer, they should also get germline testing. And of course, this is all paralleled with the cost of genetic testing for a panel of cancer susceptibility genes going dropping dramatically from thousands of dollars to now less than $250. So that's what we should be doing, but what's actually happening across the country, and there's not really any national data that's come out, but I just want to point out a couple of publications at UCSF, and of course, this is predating the guideline recommendations. You can see that despite attempts to send some patients to genetics, there always is a drop-off when you see someone in the clinic with cancer and then send them somewhere else to see a genetic counselor to have the follow-through. There was a little bit more recent organized effort from our colleagues at MSK and Penn, where they had more point of mainstream model testing, and there in a very organized academic setting, there was a testing success rate of about 90%. I'll just show you the data of what we have at NYU, and we started from scratch. And over the last couple years, we started with having a genetic counselor built in clinic, and now we have more of a mainstream model. And here we have a little bit over an 80% uptake. But I will tell you, in our health system outside of the multidisciplinary clinic, the uptake is less than 5%. And I suspect if you ask across America how many patients with pancreatic cancer actually get germline testing, it's probably less than 10% across the country. And obviously that's an important thing to fix. So just a couple of points here. We should maximize the use of the tools we have in hand, find carriers of genes that we know can increase risk, improve compliance with recommendations for germline testing in pancreatic cancer patients. And then obviously, as an extension of that, better strategies for cascade testing of relatives. Now, I've talked about what we know, but it's amazing what we don't know. And What we don't know is families that come in with a lot of pancreas cancer in the family, and this is just an example pedigree, and anybody that has a pancreas center around the country has lots and lots of patients like this, where there's a lot of pancreas cancer in the family, and you do the testing that's available, and it's negative. So we don't know what is the causative gene. And this is 85% of these high-risk families. So I've been thinking about why is that? Why don't we know things? Why haven't we made progress in there? And I'll give you my thoughts on why we don't and perhaps a strategy to tackle this. So one thing that has become clear, and if you look at all the studies out there, one of the key missing ingredients in tackling this is if you want to put the pieces of the puzzle together to find the gene or genes that cause pancreatic cancer in family, you actually need tumor tissue. And if you think about it, we actually are not very good at collecting tumor tissue on pancreatic cancer patients because most of them die and they never have a sample collected. Furthermore, we often don't know patients that have pancreatic cancer that they have familial pancreatic cancer or carry a mutation that puts them at risk. And so I'm encouraging everybody that's out there, especially centers that where we have surgical colleagues here, to try to make sure you get germline testing on your pancreatic cancer patients and then collect fresh frozen tissue because that's the best tool to do whole genome sequencing, which is going to be needed. Occasionally we can find old paraffin blocks, but doing whole genomes on paraffin blocks still has not been optimized. And then in addition to having the tumor tissue, you also need blood samples or DNA samples from affected and unaffected members of the family. And so really for us to tackle this, we need to have a systematic, concentrated focus on collecting these kind of tissues from patients who are part of FPC families. And sometimes it's with their own cancer diagnosis that you discern that they're part of an FPC family, or if they have a relevant PGV. So that's one thing. We don't have the right sample sets. Two, do we have the right tools? And just in talking to some of my genetic colleagues, what I've come to find is the sequencing technology that we use commonly, the Illumina-based technology, is all based on 200 to 300 base pair short reads. So the DNA is chopped up in a million little pieces and it's sequenced and then it's bioinformatically put back together. But there's actually new technology called long read sequencing. And in fact, just in the lay press, it came out that we have now just for the first time fully completed our analysis of the human genome because there were some missing pieces in there that were only discerned by doing long read sequencing. So this is really pretty new stuff, but I want to show you, and I wanted to point out, you can see there's just a few papers out there on this technology, but it's starting to gain a little bit of traction. Part of the issue is it actually is more costly and bioinformatically much more intensive. And so it's going to take a little time to bring it into play. But I just want to point out this one paper that the head of human genetics led me to that was just came out in bioarchives, where they did targeted long read sequencing, and they basically took a bunch of people, and these are mostly people with undiagnosed rare neurologic disease, and compared short reads versus targeted long reads. And using this technology, they actually were much more successful in identifying previously unknown mutations. And many of these mutations, but not all of them, are actually structural variants, which makes sense, right? So if you have longer reads in the DNA, it helps you to discern mutations that may affect structure. So I think this is something we want to pay attention to. It's important to realize that we still don't have the data, and there'll need to be prospective studies of different patient populations to see if this kind of technology, targeted long reads, will be superior to conventional strategies. But at least in this paper, they put forth a pathway that can be a cost-effective approach. And certainly we want to have pancreatic cancer be in the game here. Also want to bring this to your attention. You know, we have standard panels where people look for changes in pathogenic mutations, but now there's some addition of RNA sequencing to these panels, and this can provide added value. This is actually a slide I got from Ambry Genetics, and by adding RNA sequencing, it can actually improve two things. One is there's an entity called a variant of unknown significance, or VUS, and those are changes in the DNA that are presumed to probably be benign, but aren't conclusively shown to be so. And you can see, comparing the bars on the left to the bars on the right, with RNA sequencing, you can clarify those VUSs in many more patients and reclassify them to be totally benign. The other thing is, with RNA sequencing, it allows you to find pathogenic mutations between genes. And that can find new variants that are present in families. So altogether, by adding this kind of technology, it impacts panel reads in about 1 in 50 patients, which is actually quite substantive. Additionally, it could be that it's not, it's not a single gene we're missing, but that it's actually a multifactorial process. And this is an area where there's been some movement in other cancer types, breast cancer, et cetera, and polygenic risk scores, where SNPs, or single nucleotide polymorphisms, which are common in the population, may have modest effects on pancreatic cancer risk. But if you actually take all of them combined, the effects can actually be as important as a major mutation like a BRCA mutation. And if you look at the data table on the right from a recent paper, you can see if you take the first versus the, I think it was the 5th quintile, I can't actually see because of my glasses on, but if you compare those two, there's a 14-fold difference in risk. So that's actually quite substantive, and this is something we need to be thinking about. I'll talk a little bit more about that in a sec. So kind of in summary, if you look at this possible explanations for missing heritability, it could be there are a whole bunch of private uncommon mutations. And so you really have to dig into each family to sort it out. Alternatively, it could be that we haven't had the best sequencing technology, and now we have new tools, and there may be a whole class of mutations that we just missed. Or additionally, there could be significant unrecognized polygenic contribution. And so these are all possibilities, but how are we going to sort this out? And so I'm going to make a case of what we've tried to set up to get where we need to go. And this is something we started maybe about 3 years ago, where it seemed like one of the big things we didn't have in the field was a large organized cohort of individuals at elevated heritable risk of pancreatic cancer to study for the field. And so we set up this consortium called the Pre-Seed Consortium, and this is really a collaborative effort to try to get centers to work together, share data to really move the needle in this disease. We've set a ambitious goal to try to get to a 50% survival for pancreas cancer in the next 10 years by really driving this. And I'll show you this is going to grow to be the largest effort of its kind, and hopefully this platform will be enabling for our field to make the changes we desire. So what are the aims of this effort? It's to standardize the collection of demographic, clinical, imaging data, and also to have SOPs for longitudinal biosample collection for consortium sites worldwide, to generate the scientific proof that screening can save lives. For those of you who followed the lung cancer screening field, it took about 10,000 patients for that definitive New England Journal paper to come out to show that screening saved lives in lung cancer. These are the numbers we need to pull together to do this. And then, of course, by collecting data, it helps us to change standards of practice and get insurance companies to pay for genetic testing and screening. And these are things that patients still struggle with today. It also allows us to really assess risk, both genetic, environmental, and has the potential to identify new pancreatic cancer susceptibility genes, develop comprehensive risk models. And then, of course, and not lost on any of us, is a longitudinal cohort for which any early detection biomarker can be vetted. A lot of the biomarker work is done with convenience sets, retrospective samples. This is something that can be tested. In fact, biomarkers can be tested head-to-head or in combination instead of all these biomarkers being tested in silos everywhere. So this is a longitudinal prospective study. And our goal, and we have a path to collect 10,000 high-risk individuals that we follow longitudinally over time. Any site is welcome to join, but we do try to have some basic requirements for joining. You have to have your act together as a basic requirement. You have to have a multidisciplinary team. You have to have a reasonable number of patients that are high risk that you follow. And importantly, and you cannot join the consortium unless you agree upfront to share data. There's an organizational structure, executive committee, lots of different committees that you can see here focusing on important things. We have a superb genomics team, biomarker team, modifiable and risk factor team. Any project that gets put forth can be vetted by an ancillary study team, and then a publication team with the whole goal of being collaborative. And then the data is managed by Arbor Research, which is a nonprofit that serves as a data coordinating center for many of the large NIH consortia. We have active 25 sites in the US shown here on the map, 10 international sites, and between another 6 to 10 sites that are in the process of being vetted and joining. We use a central IRB, so everybody is rowing in the same direction. So this was actually an important part of what we built, which was let's capture all the data. And so there were a number of different cohorts that were established with the goal in mind that anyone who would come into a high-risk clinic would be captured. Cohort 1, as you see here, is the standard folks who would come in that are classically defined as high risk. Cohort 2 is what we call there's smoke, but we don't have a fire. So maybe they have multiple family members of pancreas cancer, but not a first-degree relative. We also capture anybody who has an ATM mutation or a BRCA mutation. So we really want to capture data so we can learn. Cohort 3 is actually people who are high risk, but they're too young to include. And there's still research that can be done on those folks. And these folks want to get engaged early. Cohort 4 are folks with a single family member, first-degree family member with pancreatic cancer. So we need to understand what happens to those individuals. Cohort 5 is if you want to do family studies to collect biosamples for people that are somewhere else in the country or the world and can be stored just for research. And then cohort 6 are pancreatic cancer patients that either have FPC or have a PGV relevant for pancreatic cancer. And that gets back to the point I made earlier about having that tissue in hand for gene hunting. Just to show how this can really have an impact, we've been enrolling patients across the consortium as more sites have been joining, and this has just been over the last 18 months. We have a little over 1,700 patients enrolled. We'll have 2,000 by the end of the year. We'll have 3,000 by next June. You kind of get the picture. If we get to 100 centers and 10,000 high-risk patients, we're going to have an incredible data source for the field for study. Also, just want to point out, as a group, we've given some thought about just how do we go about getting us where we need to go, not only at the centers I talked about in pre-seed or in academic centers around the world, but even just globally for patients being seen in community clinics. What are the basic things that should be done for individuals who are at high risk? What basic data should be collected? So this is it's on came out in Gastro if you want to look at it. I point out a few things about family history and genetic testing, but there are more general recommendations about having an organized and effective approach as a field. A couple things I've already shot out from pre-seed already. This is the pre-seed GPS study, and this is going to be a 10,000 longitudinal patient study where patients are going to get DNA panels and RNA-seq, and also all the known pancreatic cancer SNPs are going to be put on this panel to look at the impact of having that information across this large group. by having such a platform that we've built, it brings new people into the field. Some of you know me, know that I say if we can land on Mars, we should be able to detect pancreatic cancer early. We just haven't brought the right people to the table. This is an example of bringing people who weren't working on pancreatic cancer to the table. With this effort, it's called the Lattice effort. I always joke this is going to be written up as a case study in Harvard Business Review. It's bringing 4 companies and 39 centers together to work together on this. And importantly, we have experts in genomics, AI, machine learning, and Amazon volunteered to provide a free data cloud for all the data, including all the imaging for all these patients is going to be loaded on the cloud, de-identified, curated and collated for the research community, which I think is going to be a tremendous resource. And then, of course, there are additional strategic partnerships underway. So in conclusion, I think we really need to have a new strategy to go after impacting survival of pancreatic cancer. I'm going to make a case, and in addition, and I think it is important we develop new therapeutics, that we really should make a major investment in early detection. My title of my talk was The Next Era in Genomics and Pancreatic Cancer. I'm going to say the next era should be focused on understanding the whole genome, germline genetics. This is going to get us where we need to go. And I think for recalcitrant problems like pancreatic cancer, going big, having platforms like this, you heard about Precision Panc, Pre-Seed, there were other large scale efforts by our colleagues around the country, going to increase the pace and scale that we can accomplish change. I want to, while I'm presenting the Pre-Seed effort, I just want to highlight the amazing contribution of all of my peers. There are hundreds of people working on this, and it's really a collective team effort. And then my last slide, I don't usually quote people, but I like this quote from Bill Gates, which is most people underestimate what they can accomplish in a year and underestimate what they can do in 10 years. So I'm going to issue the 10-year challenge, 50% survival. Let's try to go for it. Thank you very much.
Thank you, Dr. Simone, for excellent talk. Last speaker for our session is Dr. Anirban Mehta, who's going to present virtually. Anirban is very well known to this group. He's one of our past presidents and a very eminent pancreatologist. Anirban got his medical degree from All India Institute of Medical Sciences and then did his residency and fellowship and his initial faculty positions at Johns Hopkins. And then he moved now to MD Anderson in Houston, where he is the professor of pathology and deputy division head for academic science, division of pathology and lab medicine. And also he's the scientific director for Sheikh Ahmed bin Zayed Center for Pancreatic Research. Anirban has really a leader in this area. He has published probably almost 500 papers in his still young career. So Anirban, welcome virtually. I wish you were here, but we welcome you.
Good evening, everyone. I'm Anirban Mehta from MD Anderson Cancer Center. It's a real honor to be speaking to all of you today at the Hirshberg Foundation Symposium. I'm sorry I could not be there in person due to travel restrictions. You've heard 3 spectacular talks on the topic of genomics of pancreatic cancer from Drs. Biankin, Siemian, and Wood. They're a hard act to follow, but I will do my best. So this is the building at MD Anderson where we are located on the 3rd floor. That's where the Pancreas Cancer Center is. So in the last decade, as you've heard, we've made remarkable advances in understanding the genomic landscapes of pancreatic cancer, precursor lesions, high-risk cohorts, and we're really implementing the lessons learned from these in the clinic now in terms of targeted therapies, cancer interception, early detection, etc. So there's a lot of excitement in terms of how genomics is being applied in the context of the care of our patients. In our lab, over the last decade, we've really taken the lessons learned from the TCGA and ICGC and other sequencing efforts and tried to functionally annotate the roles of these recurrent mutations that have been found using autochthonous models. And we use, and we sort of make these models because we realize that there's always a surprising twist that you can learn from these genetically engineered models that would not be evident necessarily from cell lines or other ex vivo model systems. So this is the TCGA landscape data, and some of these arrows point to models that we have made in our lab, others that are in progress. I'm going to talk to you today about one story that involves RNF43, which is another recurrently mutated gene in pancreatic cancer, and what we have found, at least part of our data, because I only have 20 minutes. So we were interested in RNF43 not only because of it being found in the context of invasive neoplasia, but also when I was at Hopkins, Bert Vogelstein and colleagues published a study that showed for the first time that this gene was also mutated recurrently in mucinous cystic lesions of the pancreas, including IPMNs and mucinous cystic neoplasms. And subsequently work by Dr. Wood and others has really cemented the role of this in terms of early pancreatic neoplasia. So what is RNF43? The RNF43 is an E3 ubiquitin ligase, and its role as a negative Wnt signal regulator was first identified in the context of colon cancer preclinical models by Hans Clevers and others. So RNF43 has a protein, has a transmembrane domain, and at the cell surface, it binds to and ubiquitinates frizzled receptors, which are receptors for Wnt ligand. And so when you don't have RNF43, you have higher levels of these frizzled receptors and increased ligand-dependent Wnt signaling. And based on this premise, actually, there's also studies in preclinical models that showed that RNF43 mutations confer sensitivity to Wnt inhibition in the context of targeted therapies. So in pancreas, there's a couple of studies that were done using established cell line models. Again, these were not done in animal models. These were cell lines. And this was a study from Novartis that used an inhibitor of porcupine. Porcupine is an enzyme that is required for synthesis of Wnt ligands. And again, remember, when you don't have RNF43, you have more ligand-dependent Wnt signaling. And so this porcupine inhibitor, LGK974, was shown to have growth inhibitory effects in the context of RNF43 mutations. And then this was a work from a group in Canada, from Stefan Angers, showing that antibodies against frizzled receptors inhibited the growth of RNF43 mutant pancreatic cancer cells, both in vitro and in xenografts. And again, on the premise that loss of RNF43 increases frizzled receptor levels on the surface. So based on these, on the sort of the body of evidence in preclinical models, there was actually a phase 1 basket trial that was done at MD Anderson using the LGK974 compound. And unfortunately, that was did not show any evidence of activity, including in pancreatic cancers that had RNF43 mutations. So it was time to go back to the drawing board, so to speak. And so we made a model of RNF43 conditional loss in the pancreas on a KRAS mutant background. And not unexpectedly, these mice ended up getting IPMN-like cystic lesions. You can see here that that cyst in the pancreas, it's got a lot of fluid in it. It almost looks like a main duct IPMN. And you also had a lot of branch ducts that were involved by these papillary lesions. So it really looked like IPMN-like phenotype. And in a subset of these mice, you went on to develop invasive adenocarcinomas, just like you have in the context of human IPMN lesions as well. So to understand a little better about the effector pathways that may be deregulated by loss of RNF43 in the pancreas, we did single-cell RNA sequencing at different stages of the disease, early, intermediate, moribund. And one thing we saw immediately in the pathway analysis was while Wnt signaling was showing up very early on, it was lost very quickly in the disease process. So that Wnt dependence that was observed in colorectal cancer models, it is not sustained, especially in advanced disease. So we think that that may be more of a colorectal cancer phenomenon, and RNF43 may be behaving somewhat differently in the pancreas. So the other thing that we noticed was very interesting in the microenvironment, and that is that in contrast to the more commonly used KRAS P53 models, or what we call KPC mice, where there is a profound upregulation of myeloid cells in the microenvironment, we don't see that. And in particular, we don't see these so-called inflammatory macrophages that are immune suppressive and tumor promoting, that are very characteristic of the tumor microenvironment in these KPC mice. So here you can see when we map out the myeloid cells early on and even in the intermediate stage, you see these macrophages, but in the actual established tumors, they are significantly depleted. So again, this is in stark contrast to what is seen in the KRAS P53 model, where this big orange blob in the middle, this is a paper from Dave Tuvison's lab, but this is from Rolf Brecken's lab. You have a lot of these myeloid cells, and they're really not there in the KRAS P53, RNF43 model, I'm sorry. So in addition to the lack of these myeloid cells, these inflammatory macrophages that is shown here. So we basically did a sort of overlap of the tumor microenvironment in KPC versus KRC, which is RNF43 deleted mice. We see this depletion of macrophages. There's not a lot of blue here, a lot of red. That's the KPC model. The other thing we see is when we look at the granulocytic MDSCs, which are also immune suppressive, they're really quiescent in the KRAS RNF43. Again, this is a feature that's very different from the KPC model. And here is sort of the summary of the immune landscapes between KPC mice have a lot more myeloid cells. They don't have a lot of T cells, don't have a lot of B cells, barely have any plasma cells. In contrast, the RNF43 mutant mice have very few myeloid cells, a lot of T cells, a lot of B cells, and plasma cells. And you can also see that they have a much longer median survival compared to the KPC mice. So why is this happening? Again, I'm cutting to the chase here. Essentially what we found through multiple orthogonal assays is that the KRAS RNF43 mutant mice don't make CXCL5. So CXCL5 is a defining cytokine that's present in pretty high amounts and secreted by KRAS mutant pancreatic cancer models, including the KC mice and the KPC mice. But when you have mice that have RNF43 mutations, they essentially make no CXCL5. And CXCL5 is pivotal in terms of attracting these inflammatory macrophages to the tumor microenvironment, and is responsible in turn for the immune suppression that is seen as a dominant feature of the tumor microenvironment in the sort of the conventional p53 mutant models. But we don't see that at all in this RNF43 mice. And instead, when you take these KRC cell lines that are derived from the tumors and you express exogenous CXCL5 in them, you basically end up getting bigger tumors, and the microenvironment now starts looking like a p53 mutant microenvironment. So essentially, you convert a KRC model into a KPC model simply by re-expressing CXCL5. So why is CXCL5 not being expressed? And again, I'm cutting to the chase here through a lot of data. It's driven by HDAC. It's an HDAC-mediated repression of CXCL5 expression that's happening, and in particular, HDAC8 that's causing this. So we've looked at the entire family, and the one that's actually causing this repression specifically is HDAC8, which is expressed at very high levels compared to the KPC and KC mice. And when you inhibit HDAC with a generic HDAC inhibitor like panabinostat or this pharmacyclics compound PCI 34051, which is an HDAC8 inhibitor, you get overexpression or re-expression, I should say, of CXCL5. So again, this happens to be driven by overexpression of HDAC8. And just a cautionary note, tangential cautionary note outside of the context of this specific model, which is that HDAC inhibitors have not had a lot of success in pancreas cancer. And I think one reason for that could also be that we are paradoxically actually making a bad situation worse, but by possibly upregulating some of these immune suppressive cytokines and bringing more myeloid cells into the microenvironment, something to think about. So anyway, we have an evolving story here as to why HDAC8 is upregulated that I'm not going to talk about, but suffice it to say that it's not a direct RNF43 ubiquitination target, because that's obviously the first thing we looked at, that maybe HDAC8 is ubiquitinated by RNF43, so when it's missing, its levels are up. That story is not as linear or simple, and it's one that we are still sort of deconvoluting here. And so I'm not going to talk about it, but it's definitely an interesting aspect of what's going on. Now let's come back to the translational implications of this. As I mentioned previously, the KRAS RNF43 mice do have a longer survival compared to either the FLOX P53 or the P53 mutant mice, almost double survival, depending on the specific study you're talking about. So is this a mouse alone phenomenon? And we actually went back to the TCGA, and it turns out that even in the TCGA, we don't have a lot of RNF43 mutant tumors, but the ones that do they do show a better prognosis or overall survival, I should say, compared to the RNF43 wild type tumors. And this is a very interesting recent paper from the Glasgow Precision Oncology Lab from Stephen Dreyer, Andrew Bianchi, David Chang in gastroenterology, showing that when they looked at mutations that correlate with recurrence or no recurrence in the surgically resected setting, the one mutation that correlated with no recurrence at a significant level, or actually approaching significant level, because again, I think the numbers are not very big, was RNF43. So when you have RNF43 mutations, it is enriched in the population of tumors that do not recur post-surgery, again, underscoring a favorable prognosis. So we think this is what's happening in terms of the sort of the two dichotomous models. In the RNF43 wild type mice, which is the KPC or KC model, you have reduced HDAC8, increased CXCL5, reduced lymphocytes, a lot more myeloid cells. This is the model that we know and we all use in our labs that is refractory to IO. But then in that small fraction of tumors that are RNF43 mutated, you have HDAC8 that's up, CXCL5 is down. Now you have sort of almost like a melanoma-like situation. You have a lot more T cells with TTLA4 and other checkpoints. You have B cells and plasma cells. And so this could be a subset that's amenable to IO with anti-CTLA4 plus minus anti-PD-1. So this is the basket trial that I hope some oncologists maybe in this audience will do, which is to run a basket trial in RNF43 mutant tumors, including pancreatic cancer, using IO-based approaches. So again, I said in the beginning that there's a lot we can learn from these autochthonous models that often proves to be a surprising twist and provides interesting insights into what we actually observe correlatively in the clinic that we would not glean from cell lines or other ex vivo models. And so I do think there's a lot of value to the effort put into making these genetically engineered mice. And in the context of RNF43 in particular, we think that this is a subset that might benefit from immunotherapy because of the paucity of CXCL5 and these myeloid populations that we know exist in most pancreatic cancers. So this is my acknowledgement. I want to particularly thank Abdel Hussein, who's an alumni of the lab. He's actually an oncologist now in Wisconsin, who did a lot of the data that I presented today. Bidut and Megan and Kemal. Kemal is a bioinformatics guru in my lab who did a lot of the single cell RNA sequencing analysis. Geeta Dangol is a former graduate student. She started the RNF43 project and a lot of the initial mouse studies that I presented. And last but not the least, very importantly, I want to acknowledge Sonia Warman. Sonia is actually an APA Foundation awardee. She has a Nature Cancer first author paper that's coming out soon on ApoBec3A in pancreatic cancer, a spectacular young investigator. And I think the APA has done really well in investing in her. They're going to get a lot of payoff from this investment. So thank you to the APA for funding Sonia. And even though I could not be there in person, I do have two members of my lab, Martha Sanz-Escoffier, who's a postdoc fellow, and Maria Monberg, who's a graduate student. Both of them have podium presentations. Martha's actually winning an award on Thursday. And again, both have travel grants. So thank you again to the APA for supporting them. And last but not the least, I want to also acknowledge Cordelia Lee, who's a brilliant high school student in my lab, and she did some really great work in terms of computational approaches to subtyping of pancreatic cancers, and she's actually also winning an award at this meeting. So there's a pretty big presence of the lab at this APA, and I'm really sad that I couldn't be there in person myself. And finally, I want to thank the person who is making all of this happen, and that's Augie Hirshberg. Augie, thank you for being such a champion and supporting pancreatic cancer researchers over many, many years. When you invited me to UCLA to speak at the Hirshberg Symposium there, I showed this slide. This was actually the last slide I showed, and it had this picture of the Energizer Bunny, because that's the way I think of you, just keep going and going. I'm sorry I can't be there to hug you in person, but you know I love you, and thank you for all your support, and thank you for making this particular session happen. And I'm happy to take questions.
Laura and Diane, you want to come up here and we'll have a question and answer session. And hopefully Andrew and Anirban can join us on video.
Yep, I'm here. Can you see me?
I'm here too.
Yeah.
Right.
Hey.
Good to see you all. Thank you for joining real time.
Yes.
Great. First question. I think the mic needs to go on. Yeah.
Can you hear me?
can hear it.
Is it okay? Well, great talk, everyone. Thank you so much. I have a couple of questions. The very first one I have for Dr. Mehta. So you mentioned regarding the CXCL5 and the usage of the specific the inhibitor against the HDAC8. Now, clearly, you already mentioned that it's not cleanest of the drugs, right? Is CXCL5 methylated? Like, can it be targeted using methylation inhibitors?
Yeah, I mean, that's a great question. The problem with all of these, DNMT inhibitors, et cetera, is that they're non-specific. I mean, you may be upregulating CXCL5, which is not a good thing to do, first of all, which is one of the concerns I have about the HDAC inhibitors as well. But you also be demethylating a lot of other things that may actually be hurting the tumor rather than helping the treatment. And so that's my concern with some of these non-specific drugs like demethylating agents or generic HDAC inhibitors. I don't know if I understood your question. Is that what you're asking? Is CXCL5 methylated?
I mean, partly, yes. The rationale was essentially that DNMT inhibitors, actually, they are more selective because if we look at the genomic profiles, hardly 100 or so genes are essentially targeted, 100 or 1,000. But we look how the HDAC inhibitors, almost 13% of the transcription profile is changed. And maybe that explains why they are not so successful in the clinic. So I was just thinking if maybe DNMT inhibitors, given their selective response, could maybe better suited for that.
I get it, but you have to remember, you don't want CXCL5 to come on. In fact, the whole point of this particular setting is the lack of CXCL5 creates a microenvironment that's conducive to IO. And so my concern with demethylating CXCL5, even if you had a very specific CXCL5 specific demethylating inhibitor, which doesn't exist, but suppose you had it, you don't want to go down that route because you're actually going to create a microenvironment that's going to not be conducive to IO. So you want to keep that CXCL5. In fact, if anything, you want to reduce CXCL5 levels, and which is what's being done actually with inhibitors of CXCR2, et cetera, the receptor inhibitors in pancreatic cancer. So you want to actually ablate the effects of CXCL5 rather than try to re-express it.
Dr. Wood, I had a quick question. Is it known whether you can detect the KLF4 in the blood or something? When you mentioned that it can be potentially be an indicative marker of premalignant, well, in the panels essentially, can it be detected in the blood, maybe as a strategy for the diagnostic purpose, or maybe?
Can I ask you to introduce yourself? Sorry, who are you? I'm sorry for the remote folks here. If you can introduce yourself.
Absolutely, sir. My name is Somnath Pandey. I'm a fellow in the University of Miami under Dr. Saluja.
Yeah, so KLF4 in the blood, I mean, I think there's no reason assay-wise you couldn't. It's a hotspot. You could detect, or you could design a circulating tumor DNA assay for it. But since it's in the IPMNs, right, the IPMNs aren't going to be releasing their DNA into the blood because they're non-invasive. So I think as far as using it in early detection, you'd have to do it in cis fluid because it's, for the most part, not present in invasive cancers and only in the precursors.
Thank you.
Thank you.
Good evening, Shaunak Majumdar from Mayo Clinic. My question is for you, Dr. Wood. Fascinating talk. We and others have shown that in surgically resected IPMNs, if there's high-grade dysplasia, then that's a predictor for future cancer in the remnant pancreas. And we know dysplasia in IPMNs is multifocal. So the question is, does the hatching of an egg somewhere in the pancreas change the mutational landscape of the remaining unhatched eggs? Is that something that you have intentionally looked at, and maybe you have, if you can share that with me?
Yeah, I mean, I think that's a great question. And I think we need to, when we think about these precursor lesions, we need to stop thinking like lesion autonomously, as you mentioned, they're often multifocal. There's often with IPMN patients, even with PANNs, there's often so many of them in the pancreas. And we've shown that when you have an IPMN and a cancer that occur together, like 1 in 5 times are completely unrelated. And so I think whether having a high-grade IPMN in one region is just a marker of whole pancreatic risk, or if there's something that high-grade IPMN does to put the rest of the pancreas at risk, I think we don't know that. But I totally agree with your point that we need to be thinking at the whole pancreas level rather than at the individual lesion level.
Yeah, and as a follow-up to that, I really enjoyed seeing the modeling of the PANINs. Is PANIN 3 as uncommon as we think it is?
It is, yeah. And I mean, especially in the absence of invasive cancer, I think a lot of the studies that have studied it in the past, when you have it in the setting of invasive cancer, it's actually cancerization of the ducts. So it's often pancreatic cancer that's grown into the ducts. And I think when you have it in the absence of invasive cancer, it's far less common.
Thank you.
Yeah.
Thanks.
Hi, I'm Mike Walker from Panbella Therapeutics. I had a couple questions for Dr. Siemian. I wonder if you could maybe comment on the state of the art of circulating tumor DNA in pancreas cancer and the possibilities for partnering with pharma as they start their studies and maybe use the centers that you already got at Perceive.
The question is about circulating tumor DNA and as a biomarker for the early detection of pancreas cancer. And clearly there's a lot of work in this field. But if you look at the data that's been produced so far, it in and of itself hasn't been sufficient really for early detection of stage 1 pancreatic cancer. Now, that doesn't mean it can't be a useful tool, and one can envision it could be useful as part of a panel-based strategy. I would encourage companies like yours to think that way. Will it be best partnered with other biomarkers, addition of hemoglobin A1C, CD19-9, et cetera, because you could envision that perhaps you need a panel and not a single read, and that'll take the work of a partnership with the private sector and an entity like Preseed. And yes, there's no issue and problem with partnering, but I think there, as with all biomarker development, there has to be a series of steps of using certain sets of samples to get to the point where you want to use, reasonably precious longitudinal samples to make your case. And when if you need such samples to get to that, which a lot of companies do, you can certainly reach out, and there are many centers that are part of this, and actually centers across this room that have such samples collected. Stage 1 pancreatic cancer, disease controls, diabetics, people with obstructive jaundice, people with pancreatitis, all those like critical biomarker questions and controls that need to happen to, build a effective biomarker. So I think it's an exciting time. There's a lot of interesting biomarkers out there, and even some of them get released for clinical use before we even know how to use them. So I would encourage companies like yours to partner with academic colleagues to figure out how to best position your biomarkers to be most effective.
I actually was thinking about it in the opposite way, in that Proceed could partner with the pharma companies to get blood and tissue from the patients who are enrolling in the studies.
Sure. I mean, I certainly, there are lots of ways to collaborate, and all avenues should be open. I totally agree.
Thank you.
I had a question for Dr. Biancan. What you presented was really exciting, and I just wanted to hear a little bit more about the timeline. Did I hear you right that you're fitting this up to be actually, to start up in January? Is that right?
No, so the January is the full diagnostic strategy for all solid cancers. So one of the things we've been trying to do is democratize the current model of centralized molecular testing. So we have the foundations, the Tempuses of the world. The KRAS is delivering these tests in a centralized model. We believe that the future model is more about a democratized setting where individual health services can deliver these tests. And I think that we're doing this for Scotland and the UK. We can see that companies like Foundation have now kitted out their models. We have PGDX kitting out their models. etc. So kitting out their tests so that they can be provided in a way there where health services can deliver them. And so the way we can do proper drug development in the future is that we're not tied to a centralized model of molecular testing. We need molecular testing across the population, across health services, where as part of health service provision, we have molecular testing that allows us to actually understand what the molecular pathology of our patients' cancers are, so we can build and understand how tumors evolve, how tumors change with treatment, even what do the tumors look like in pancreatic cancer. We don't even understand what tumors look like for the patients that come into our clinical trials. So we need a broad-based healthcare system to do that. And so we've pushed our way forward to enable easy access to the wet chemistry to be able to deliver this. And so that is the way forward, as far as we see, is that all health services can deliver patient molecular testing for their cancers so that we can enable accelerated drug development for the future.
Well, this has been a terrific session, a lot of new information brought forward, and we really appreciate, again, Aggie Hirshberg's support. Aggie would like to say a few words.
And while Aggie is coming, we really appreciate particularly Dr. Biancan, where it's almost midnight for you, Andrew. So thank you for joining us that late. And we miss you here. Hope next time you'll be here, and for you also, everyone. We look forward to seeing you soon. Aggie?
First of all, it was a fantastic session. Dr. Biancan and Dr. Wood, it's very nice to meet you. And from now on, we're best friends. So that's done. So Diane, Ashok, Joe, and Anirban are all board members of our scientific advisory board of 12. Our boss is Bill Go. Is he here? Our boss is Bill Go. We all listen and respect the scores. And we're grateful for Dr. Go for keeping us organized. We started funding, or the foundation started funding probably 2005. So in this, from 2005 to present time, we've funded over 100 grants at 46 various institutions, which is pretty good. And when we first started, as Dr. Go says, we had no manpower. So we had a lot of young people apply, and many of you are in the audience right now. And now we have the manpower, and now we have the information. And what I love hearing so much is the word collaboration, because without collaboration, we might as well all go home. Anyway, loved it. Is Miklos here? Because Miklos is also a board member. So it's not moi that picks the projects. It's a wonderful, wonderful, brilliant group. So thank you for being with us.
All right, thank you to all of our speakers, for everybody participating, and if you can, and please join us for the reception, which just starts in about 5 minutes.
That's true. We have a reception to honor our president. So both Dr. Rustigi and Dr. Hines. So please join us outside in 5 minutes. Thank you.